As the climate warms, plant diseases are expected to increase around the world and across many crops. For farmers, that means disease pressure on their crops and a risk to their yields, and therefore a reliance on costly tools like pesticides or leaving fields unplanted for a whole season. But plants are not facing disease alone. Every plant root is surrounded by a community of microbes, including fungi and bacteria. Some of these microbes are harmful pathogens that cause disease, while others are beneficial microbes that can help plants to access nutrients, strengthen their immune systems, or fight pathogens. In our lab, we study these microbial communities on wheat roots. The big question we ask is, can the wheat that we plant today shape the soil for what grows tomorrow? The image on the screen shows 2 experimental wheat fields in Oregon. Each small rectangle is a different wheat cultivar, or a group of plants that have been bred for specific traits. The field on the left shows a first year of planting. As each cultivar grows, its roots are interacting with the microbes around them. The field on the right shows a second year of planting. We plant new wheat into those same plots to ask whether the cultivar grown the previous year changed the soil to affect the subsequent crop. Some of those differences you see, especially on this right side, are disease symptoms. In other words, wheat cultivars have left behind a microbial legacy in the soil. Here's one way to think about that. Imagine a neighborhood. The people who live there shape the environment. They influence what businesses move in, what resources are shared, what problems build up, and what kind of community the next residents will inherit. Wheat cultivars do something similar, but underground. Some cultivars may be more susceptible to disease and therefore leave behind more pathogens, creating a disease risk for the next year, while others may leave behind a buildup of more beneficial microbes, creating a healthier starting point for what grows next. That is what we mean by a legacy effect. Our results show that the cultivar planted in year 1 can influence how well the next cultivar performs in year 2. If we can identify which cultivars leave behind healthier microbial communities and eventually which genes help them do that, we can give farmers a new tool for managing disease. That could help reduce disease pressure and support soil health, which can lower reliance on pesticides and reduce the need to leave fields unplanted. Instead of only asking, which wheat should I plant this year, farmers could also ask, which wheat will improve my field for next year?